Yes. Good evening, everyone.
Good evening, sir.
So,
Good evening, sir.
yesterday we have seen CRD, FRBD. Today we will complete that topic with LSD, then alpha lattice, split plot, and calculation of blobs and blue. Yes, that the field topic will be over. So once I will revise that CRD and FRBD yesterday's. So yesterday I have shared you the data for complete randomized design and factorial RBD. Along with that, I have shared the script for how to convert the AppMap file into numerical data and uh, along with it, one script for PC. So just you can see, I will share this script. Yeah. So yeah, so this is our CRD data. First column genotype, second treatment, third replication, and from fourth column onwards, these are our 13 traits. So this is total data recorded in complete randomized design. And simultaneously, this is the script. So first you have to make sure all these packages are installed in your system. After that, simply you need to run the library command for all. Then just set the working directory. After that, just import the data with the command line. So here data is imported and then you need to proceed one by one. Then here you need to define the traits. Here just you need to adjust this place. After defining of the trait, then we need to build the model and we need to go for ANOVA. Something. So after defining the trait, then you need to proceed for this script. Soul is single only. Where is the mistake? Where is the error? Ah, this is the error. Okay. So what is the error? Why this error is coming?
Capital W is replaced with small w.
Huh? Come again?
In this case, TW is not given with the grams, with the units in bracket, GM in bracket.
Yeah, actually in data units are not there, but in command we have given units. So because of that, that error is there. So now it will run. Yeah. So yeah, Raj is there. Hello, Raj.
Yes, sir. Yes, sir.
Now fine.
Yes, sir. I understood, sir.
Okay. So this is CRD. Here you will get the graph. That is fitted value against residual. So what is mean by fitted values? Yeah, fitted values are which values? And what is residual?
Sir,
What is fitted
residuals means
values and
left out value.
huh?
Sir, residuals means left out value.
Actual value minus prediction value is residual, sir.
Okay. And fitted?
Fitted value, and fitted value means I think that actual value is equal to our expected value.
So fitted values
And
are the predicted values by the model. So if you will see the command line, here we have set one model. So see here we have build one model. So fitted values are the predicted values by this model. And this data points, all these points are your observations. and actual value minus predicted value, you will get residual. And here on right side, it is QQ plot. So this here we will see whether the data points are distributed normally or not. So likewise, you will get for each and every trait. So these are the treatment means for each and every treatment. And then here, I think text file will be there. Yeah, these are all the plots at a time we have exported. Then in this analysis, we have got the ANOVA table and then LSD. So what is the purpose of LSD test? This Fisher LSD. Yeah.
Comparing the means.
Mean comparison.
Yeah. Comparison of treatment means. From that means we can conclude like which treatment is significant and which treatment is non-significant. So apart from this, you can go for PCA analysis plot diagram with this density plot with this CRD data. So that all visualization part you can complete.
Excuse me, sir.
Ah.
With the QQ plot, what can we say in this CRD analysis? Like most of the line is not um, on the. So when we see like yeah, the QQ plot, not so some of the genotypes or yeah are on the line, but most are like diverging from the line. So what
So
can we
what
say?
is the meaning of that divergence means what?
What I'm thinking like is the not going in the direction of the population, the majority of population, like the
So what
traits.
is the what is our ultimate objective of drawing this QQ plot? Why
Mm,
we are drawing this?
why to check the population distribution, like how it's going?
Yeah, so this central axis means our that is normal distribution. And now if you will see, up to some extent, our data is showing normal distribution, right?
Yes, sir.
Yeah, but after some point, suddenly the data points are starting to divert.
Yes.
Yeah, so why
So
is
we
it so?
so we can say like these are the population, they are diverging. So maybe they they are good for selection, or what can we decide from the
What about the outliers? Here, some points, some data points you can see
they are
separate.
the outliers. Okay, yeah.
So
Yes.
these points like somewhat these points here, or you can say in this residual versus fitted plot.
So those genotypes, they are on the normality line in QQ plot, and if they are in the fitted value, then we
So
go for
simple,
selection.
so simple is that this QQ plot is showing like whatever the model we have applied.
Yes.
So we are checking whether this residual is following normal distribution or not. Okay, this is the first
These are
first
not the
concept.
fitted one. These are the residuals one in okay QQ residuals.
Yeah, here we are seeing the distribution of residuals.
Okay, okay. And the
Residuals
residuals
means
are those
our
that is not fitted in the model
actual,
what we designed.
yeah, actual minus fitted.
Okay, sir.
Actual minus fitted.
Okay. Thank you.
Whatever the data points will fall in the normal distribution or close to the normal distribution, that data points we can conclude like those are following the normal distribution.
Yes, sir.
And those are present at the extreme level, so you can name them as the outliers. Might be those points are the outliers because they are showing the extreme values,
So for the
as you can see at the top as well as at the bottom.
okay, sir. So the final outcome, finally, we will select the lines for further analysis, those who are fitted and we can take some from those follow the normality in the
So
residuals.
whatever this middle is there, the following normalities. So in that data, if you will see here, everything is there.
Yes, sir.
Yeah. So here for each and individual treatment, this all the analysis is there. So from here, you can conclude that just you can correlate that values and plots. So this is CRD, and in case of FRBD, yeah. So here we have for 3 factorial experiment. First is genotype, second is treatment, third is salinity, and these are the 11 observations. So as we have already working directory is set, so no need to set it again. So in FRBD, we have 2 different scripts, one for analysis and second one for graph. So first we will analyze the data. We will upload the data through command line. What happened? What not exist? Okay, so then here, if you are annotating the file with some different name, so you need to edit these names wherever it is there. Here we are annotating it with data. So after annotation, just simply follow the command line here. We need to define the traits. Then now here we are creating the output files and plots. Yeah, so these are the interaction plots first we'll get for all the 11 traits. And along with that, you will get the analyzed result. Analysis of variance. And then afterwards, this second command for graphs. You can start from in between. I think I have shared the command for export to export all these graphs, na?
Yes.
So likewise, these graphs will come, and for interaction plots, you will get in one PDF. So if immediately after analysis PDF is not there, just refresh the folder, you will get these files. Someone asked me in the morning.
Sir, I actually again tried for the same, but what is happening is now when you open the interaction plots file, you have 33 pages, means 33 different plots are there. But in my folder,
Ah,
it is just 11. I don't know why is it happening. I'm not getting these charts. Whatever you are showing now, the interaction
ah,
plots, I'm not
ah.
getting these ones. And
Why?
there is showing no error while running the commands, means there is not showing any warning or error, anything. It is working. But I don't know why is it happening.
Yeah, script is working properly, but plots are not coming.
Yes, exactly.
Mm,
These genotype into treatments and all the interaction plots are not there.
yeah, here in my case, for every character, 3 plots are there. So
So
that's why total 33.
this is not coming in my case. That's what
So
the
just
issue is.
do one thing, save that FRBD file in some different folder and change the directory.
Okay, sir.
Change the
I will.
directory or just in case, check for the updates.
Okay.
Check for the package update or RStudio update.
Okay, sir. I will do that.
So
Because
if it
this is the
is,
only thing now left. Because I tried two or
yeah.
three
So just
times. Same happened.
no problem. Just try it. Just check the updates. If it is updated version, so like this, it will come.
Okay, sir. I will try.
Yeah. And for package update, you need to check from here.
Okay, sir.
Yeah. So from this FRBD data also, you can go for PCA, chord diagram, density plot. So representations you can cover. Yeah. So now next part is split plot. Yeah. Anyone, what is mean by split? What is split plot design? Anyone is
Sir,
working on split plot?
sir, if the soil fertility gradient is goes in opposite direction, we can go for split plot, sir.
Okay. So here you can see the data file. 3 replications, main plots, subplots. So what is this main plot and subplot?
Main plot could be some specific treatment, sir, and the subplots are maybe the varieties.
Yes, sir, I think that exporting graph command, I think it is there in WhatsApp group. Is it? Yeah, just check the WhatsApp group, sir. You will get it. So yeah. So here, this is the split plot data where you can see the three replications, then main plot, subplot with six different characters. Six different characters. And where is the command? Yeah. So for split plot also we have two different commands. That is one for analysis and one for visualization. So generally, can anyone tell me within if we have this split plot experiment, generally we use more number of genotypes or one single variety with different treatments? What will the kind of split plot experiment means what? Like RBD, we used to randomize 20 treatments in three replications. So likewise, what is actually split plot?
Sir, generally few varieties for different treatments are used.
Treatments.
Yes, anyone else?
Single variety with many treatments.
Yeah. So actually at a time we can go for in one variety, we can go for two different levels of treatments. Like in main plot, we can go for irrigation level, and in subplots we can go for nitrogen level, and just we can analyze that split plot in data. So here likewise three replications in main plots, irrigation levels are there, and in subplots nitrogen level, and this is the data. So in script, just check whether you have already these three packages. So just first set the working directory, proper working directory. Then run the library. If any packages are not there, just install it. Then import the data. Then conversion of factors, that is 3 replication, main plot and subplot. Then just define the traits. Because we have defined the how many traits and which are the traits. And then the final step, running of the analysis. And after that, we need to save all those outputs. So after completion, you will get the output file in the working directory. Text file with already having the annotations for significant, non-significant levels for all the traits. And here, the graphical representation. So see, these 3 are the subplot factors. Then these are the main plot factors, and this is the observation. So likewise, graphical representation is this, and in other way, the second script, R script for split plot visualization. That is representation of mean data. So
Sir, can you again show the script for split plot, sir?
yeah, I will repeat 1 minute. Is the error. Yeah, so first see, there are 2 files. So see the visualization of split plot. Let's see the box plots for number of parts across the treatments. So these all are the main plot into subplot. Then these are the observations. These are the I1 main plot, and these are the second. These three are the second main plot. Likewise for all the traits. Yeah. So then here main plot, subplot factor, interaction for yield. So this type of plots are there for each and every trait. Then at last, you will get this combined one. Trait-wise interaction plots. So along with analysis of variance, so this type of plots will be there, and this type of plots will be saved directly into your working directory. All the plots. No need to save it independently. So initially, in first command, you will get this type of interaction plots. And in second command, that is visualization, you will get this type of plots. That is box plot and interaction plots for all the traits across the treatments. And this is the trait wise. If you will see the analysis part, here you will get the ANOVA. Main plot, subplot, residual, everything. Trait wise it is there with significant, non-significant annotation. So each and everything. Apart from this type of graphical representations. So these graphical representations are there. So this is your split plot design. So any errors are there in this? You have not faced this error? Yes. This melt function.
Yes, sir.
So just follow what I did. Just run the library reshape 2 and then continue library reshape 2 and then continue with the commands. If you will get the error called could not find the function melt, so run library for reshape 2. If it is not installed, so install it and then run library and then run the analysis. I think reshape 2 we have installed already. If it is not there, install it and run library and then run analysis. Yes, Ragini.
Sir, I ran the first script for script for design, but I'm not getting any output file.
Your voice.
Sir, I'm not getting any output file. I have run the whole script, the first script.
Output file. Just refresh that window. Right click and refresh it. One text file will be there. So after running the complete first split plot command, what is the last line in black color? Is there any line printed in black color at last?
No, sir, it is showing only plus sign. Should I send you the screenshot?
No, press escape and run that command again. Because if you have run the command correctly, plus sign will not come. Like this greater than sign will come. If you have pasted copy command in between, then only plus sign will come. I will show you. So see, this is the command. Suppose I will copy from here. So actually this command is starting from here to here. But if I will copy from, I think it is not visible. Yeah, so see, actually this command is from here to here. Okay. But if I'll select it from here to here only and then copy, and if I'll paste and enter, so this type of plus sign will be there. So is it like this only, na?
Sir, I have performed only first script until now, not the
Yeah, first script
visualization.
only, but I am asking about this plus sign.
Yes, sir, it is coming like this only.
So it is not completely run. You have not selected the complete command.
Okay, sir, I will do it again.
Yeah. So in this split plot, there are lots of brackets are there. So just make sure you have selected the command, total command line. So this graph, initially you will get one text file. After in that simultaneously you will get this type of graphs, not this type. And this type of graphs you will get. And in second command, you will get this type of graph with this box plots. And output file like this for each and individual trait. So that is what this split plot design. So anyone is having any more error in this split plot? Completed up to this?
Yes, sir.
Okay, then our next is split plot is over. Yeah, then our next is LSD. LSD design. What is LSD?
Let me for design.
When we'll use this? When we use Latin square.
So when the number of treatment and replication both are high.
Yeah, anyone else? When we will use the Latin square design.
Treatment and replication is equal.
Just you can open the data. And try to get idea from that LSD. Yeah, Purva, it is square design, correct?
Number of rows and number of columns.
That's what. Yeah. So as it is a square design, so we'll test the genotypes across rows and columns. That is row wise and column wise variability for each and individual treatments. So likewise here see 4 rows, 4 columns. So total number of plots?
16.
So here number of plots are 16.
Sir, screen is not visible.
Sir, your screen is not visible.
I stopped. Why I am stopping this? Yeah. So see 4 rows, 4 columns. Number of plots are 16. And these are the 4 genotypes. And these are for data for 4 traits. So likewise, arrangement you need to do for square design. Here we are testing row wise and column wise variability or variation. Here it is not like we don't have many number of treatments. So single treatment but in square design. So across rows and across columns.
So in this all the split plot and LST, we are checking the heterogeneity of field.
Yeah, experimental designs, each and every experimental design that heterogeneity will be there, but that criteria are different. Like in split plot design, if we are giving some special treatment with the in our data, we have given the salinity environment. So salinity, that different levels of treatments of physiobiochemical agents and genotypes. So this, on the basis of these three factors, we have split the plots. But here we have only one factor, that is one treatment. So because of that, we are considering the variation across rows as well as across columns. So rows and columns means in that square only we are making small plots. So that's why 4 rows, 4 columns, 16 plots, and 4 total genotypes are 4 and characters are 4. Yeah. And this is the script. Here I think plots are not that much. Single script, I think. So here again same thing. I will clear this. So again here we need to start with the library. Then we need to import the data. So here if you can see, the data import command is different I have given. After setting of working directory, then you need to create one output folder. Then we need to define the structure, in what structure we have provided the data. Then similarly again trait column. Yeah, and here see, this is the single command in single bracket. If you will see, this bracket will start from here and at last it will end here. So if you will copy it from in between, so you will get the plus sign, that as it is after complete command, this analysis will start. Yeah. And in this output file, you will get graphs and Excel file for each and individual character. So likewise, again here QQ plots are there. Then this residual versus fitted. This QQ, and here we will get directly Excel file for each and individual trait with LSD with ANOVA and LSD groups. So how we can give this significant, non-significant here? Yes. Anybody remember that?
Sir, in P value, stars will be there, single star, double star.
How to give? During stability, we have given directly through one code.
Sir, after adding one column near the F value, then we have to put the
Okay.
codes.
Yeah, tell me how. Where is that code? I need to search it. 1 minute. I will search that code. Where I kept. If you have that code, just try it. So yeah, how to give? Tell me. I have added the column. Next,
Insert module.
from where?
Sir, first you will have to press for Alt and F11 together.
Hmm. Insert module. Then copy, paste. Whether here we need to make any change?
Sir, your screen is not visible for code. It's just Excel sheet.
Yeah. So here I have pasted. Now here either we need to make any changes or not. Hmm?
Sir, the file name.
So say this. So here we need to give the file name.
Excuse me, sir.
Hmm.
So by just directly seeing the F value, we cannot say whether it is significant or not significant.
How?
So like for F value at 0.05%, the values.
Where are the values? 5% value, 1% value. You need to see the table, na?
Yes, sir.
That one way is there, no problem. You can that convince that one way is there. Then something here problem. Heading. Yeah. So here automatically it will come. No need to add the column. At last it will come.
Excuse me, sir. Could you please just repeat one more time and share the code on WhatsApp? Possible.
Code already shared to you long back during our stability time. So just here you need to give the sheet name. Now it is correct. So like this. Significance level you can assign.
Wait once again, sir. Sir, what did you press that this name module one came, sir?
So just go in flashback during the time when we
Yes, sir.
used to do stability analysis.
Sir, function alt and something was there, sir. I don't remember third button.
So this thing is firstly for advanced version laptops function alt and F10.
Okay, sir.
10 is correct now?
It's 11, I guess. sir.
F11,
Ah,
sir.
F11.
I am also getting confused with different systems. Yeah, F11. Then in module, here you need to paste the code, delete this heading, and here you need to type the sheet name. Then just close this. Then alter plus that whatever is that function. So like this, significant levels will be there.
Excuse me, sir. I think I didn't save the script. So if possible, you could
Script
please.
you can download from WhatsApp group.
It's on WhatsApp.
Yeah. See this, the name, code for add significance level. If you will go through this files here, everything is there. You can download easily. So likewise here you can give the significance level. For each and individual thread. And then as I told you, the graphs. So these are all the graphs for LSD. So this is all about LSD. This is residuals and fitted. Yeah, so after this it's fine.
Sir, after pressing, sir, after rewriting ME Nova sheet 1, what we have to do after that?
Close everything. Don't close Excel file. Close that whatever that editor is there. Then Alt function Alt F8. Select the module and run it.
Sir, I'm getting error in adding the significance level.
To make sure whether you have deleted the heading or not.
Sir, I have deleted it, but still it's not happening.
So see, this is the ANOVA, correct? So first I will press Alt 11. I will go to insert. Screen is visible now?
Yes, sir.
Yeah, then module. Here I will press the paste the code. I will delete this heading. Here I will type the sheet name, that is sheet 1. So this name typing is correct.
Yes, sir.
Capital S, and there is a gap in between sheet and 1. Then close this. Alter F8. Here select, it is already selected. Just simply click run. Levels will come.
It's done, sir. Thank you, sir.
Mm.
Sir, in which case we will be getting 2-star and 3-star, sir?
Huh?
So like I'm getting for plant height I'm getting 2-star.
Ah,
So like in which case we'll get 2-star and in which case we'll get 3-star?
these are the significant codes.
Okay, sir.
And simultaneously, I don't know whether you people are aware or not. One GUI based software is there called Grapes. I think developed from Kerala Agriculture University, if I'm not wrong. It is also it is still in development stage, but that is a bit easy. It is based on R only, but that is somewhat easy that you don't need to work on that much of coding. Still, not everything is there. Only basic analysis is there at present. If I'm not wrong, it is developed from from Kerala University scientists. Just once you can check whenever you will get time. That is called as grapes. Okay, so this is LST. Then our next is alpha lattice. I think it is having some error. Okay, I will share that data later on. We'll cover it. Yeah. So the next is estimation of bluffs and blue. So what is bluff?
Best linear unbiased predictions.
Okay. And blue?
Best linear unbiased estimators.
Okay. So first is best linear unbiased prediction, and second is best linear unbiased estimator. So we have to calculate this bluffs and blue from the raw data. So what is the meaning of raw? Raw data means what?
Ungrouped data.
Okay, whatever the data we will record directly from field. So from that data, we need to record this bluffs and blue. So now first we'll see what is the difference between this bluff and blue. So actually, one is the predictor and another is the estimator. As we are saying blue, there is best linear unbiased estimation or estimator. So this blue will estimate fixed effect. So fixed effect means that is your treatment means, then environmental effect. Okay. So this blue will follow linear model. So it will assume that whatever the effects are there, all effects are fixed. So likewise, similarly, this bluff, it will predict the random effect. So that's why the name are given: estimator, fixed effect, prediction, random effect. So this bluff will follow mixed model. So mixed model means it contains both fixed as well as random effect. So random effect means you can say like your genetic potential of genotype, that is breeding value. So just here we will use this for demo purpose. We will use this TRD data. From this data, we will calculate the bluff. Okay. So just try to run the command with this TRD data for bluff and blue for both. Just clear this everything. First go for bluffs, or you can start with blue, no problem. Make sure to set the working directory. Then import the data. Then just ensure this both genotype and treatment as the factors. Then define the trait column. Go for blocks. So then up to this fifth step, that's why I have given the names for each and individual trait, and finally you will get this in Excel into your working directory. Yeah, see the CRD blobs. You see here, for all the observations, these are the blobs. So as if you will see the raw data, some data is missing. Actually, it is missing, but still this model predicts that data. If you will see this, every in between you will find that each and every value is same. So this is that in that case, it will make a groups. This all are the predictions. And then simultaneously it will go for blue. This is just I am saying taking demo. Just you can, if you will have any genotypic raw data or any raw data, you can go with it.
Sir, in step 3, that 4 and code is written. That means from fourth column, data is going to take place. Analysis
What?
is going to take place. So in step 3, 4 and code was written. So from fourth column, analysis will take place, na, sir? In the script it was
Yeah,
written.
because this first three columns, if you will take analysis, it will give bluff for genotype number also.
Okay, sir, understood.
So this is just I'm telling you the structure, how to calculate.
Excuse me, sir. What is the significance of this value, like for breeding? Blue was the analysis we did for CRD data for blue. So how can we use this estimator value for selection and something?
So actually, what is the ultimate objective is, so as these bluffs are the predicted random effect. So most probably in plant breeding, we are predicting the breeding values or genetic potential of the genotypes. Okay, so that these estimates will tell us the genotypes which are deviating either positive or negative from the population mean. So in that way, it will be useful for us for selection. And simultaneously, these blobs we are using in molecular breeding also for association mapping purpose. What we use the PF phenotypic data for association mapping, those all are the blobs. They are not the raw data. Those all are the blobs for the accessions. So it will give us the accurate ranking.
Yeah, but in this analysis is showing the same value for.
No, that's what initially I told. This is just a demo because this is the different. Somewhere it is missing.
Okay,
This
okay.
data is, yeah. So if you have another data in this way, you need to calculate. Because I have searched the raw data, but in my laptop, everything is messy. So if I will find my data, then I can show you.
Sir, so the further analysis will be based on this data only, not the actual raw data.
No. Generally, if you have recorded some field observations for molecular data, so first we are not using the as it is field data for analysis. First we'll calculate the blobs. And then that blobs we will use for association mapping. Or in other cases, as yesterday Dr. Mukherjee told, in the industrial people, what they will do, they will calculate directly blobs. So from that blob, they'll come to know which genotype is performing outside the population limit. Am I correct, Dr. Sir?
Yes, sir. Yes, sir.
Yeah. So for that, generally now see, industrial people will not waste much time in screening and continuous screening. They are the business people, industry people means. The time is very precious for them. So that's why they also started to go for these blocks. So they will select the genotypes. Those are performing beyond the limit of their whatever the population, beyond the limit of that population mean. So for that selection purpose, they will go for these blobs. Instead of screening for multiple season and then from pooled data, they will select that genotype. So instead of doing all these things, they are following the blobs.
Sir, how effective it will be for the actual field quantitative data?
Actually, see, the name itself indicates basic linear unbiased predictions. So see, we are doing association mapping. So while doing association mapping, we also don't know anything. Am I correct?
Yes, sir.
Means after map identification of markers also we don't know whether that marker will work in certain crop population or not. So that's why that concept is unless and until validation, we will not do validation, that markers is of no use. So for that purpose, one validation concept is there, that is competitive allele specific PCR. CASP marker, KASP. I think many of you are aware. So that high throughput genotyping technique used for that validation, and after validation only you can say like these markers are working. So it is everything, this is based on prediction.
Sir, for augmented, the script is same or it will be different?
Yeah, that structure is same, sir. Just you need to edit wherever is necessary. That is the trait column, file name. Okay. So just the structure is same. Just आपको वो जहाँ जहाँ पे जरूरी लगता है वहाँ वहाँ पे आप एडिट करते जाइए.
ठीक. Okay.
So this you can change the scripts based on your objective. Just the bottom line is don't change the structure of script. Inside that structure you can change it.
Yes, sir.
Then similarly, blue is there. That is best linear unbiased estimator. So in the same way, just calculate this blue. So generally, in alpha lattice, we'll go for this bluffs and blue. Alpha lattice augmented wherever we are using more number of genotypes. But wherever we are testing more number of genotypes, there we'll go for this bluffs and blue. So now in this case, these blues, it will give the adjusted mean of genotype across replications. Or if you will say, if you have tested the genotypes at multi-locations, so across environment it will give. So like here, that outcome will be, you can identify the genotypes, which genotype is better in which environment. And in case of blobs, if in some cases you have unbalanced data, so we'll say like environmental error or some experiment, while putting experimental design there, somewhere error is there. So that error, this block will remove. So after removing that design or environmental errors, you will get the true genetic potential or true genetic value of that particular accession with respect to that particular trait. So this is how these two concepts will work.
So at a time we can perform both, sir, blob and blue.
So actually, generally, sir, we will go for blocks only, either blocks or blue.
So when will we perform this blues, sir? Blob we can do at normally we are doing blobs, mostly blobs. But
so actually, the decision is based on like if you have to take some decision like selection of genotypes, then from BLUP you can select that this genotype is performing better in this condition. But when you are going for fixed effect, so then for a fixed effect, then in that condition you need to go for blue, because this whenever any unbalanced data is there, so this blue will not able to handle that, but BLUP will handle that unbalanced data. It will reduce the environmental error and it will handle the unbalanced data. But in case of blue, it will not take that, it will not able to handle. And as far as your G into E, so this G into E, this BLUP will account for that interaction, or if you will say that correlation. So because of these reasons, will most preferable is BLUP. So we'll select from blue, we'll select the genotypes from whatever we are observing, that is observed performance. But from BLUP it will remove the environmental error and if environmental and design errors, if any, and after that it will give us the true value, true genetic potential. So from that you can make decision. But in blue, just you can make the comparison in between the genotypes from the observed performance. So that will depend on your objective. Like generally, if any agronomy people are here, so generally they are comparing different treatments. So in that case, you can go for blue.
Sir, as now you said that if we have to test different treatments, we can go for blue. So if I'm having,
Comparison, I am saying comparison.
yeah, for comparison only. So now if I have some chemical with different doses, these two things are fixed and I have to test their comparison, or I have to make comparison which is best out of them, I should go for blue. Is it correct?
Yeah. For comparison, you should go for blue, but for if you want to select, then BLUP is there. For decision making, BLUP. For comparison, blue.
So if a genotype is not there, but if there are different chemicals or treatments, as you mentioned now, we can go for BLUP for selection purpose, which is best.
Yeah. Yeah.
Okay.
So what did you say, sir? For comparison, we use blue, and for so for what purpose you use BLUP? Hello?
If you want to make any decision, then you have to go for BLUP. And when you want to compare different treatments or different genotypes only for publication purpose, then you have to go for blue.
Okay, sir.
So, except alpha lattice, we have completed. Alpha lattice is having some issue. So if you have some different data, so just you can go try this calculation of flux. But here, just you need to edit it. Don't use as it is, because here you will annotate it, but here the name study genotypes, treatments, this column number, here you need to edit. Just while using, don't forget to edit these commands for all. And maybe by tomorrow you will get this everything in one Google Drive link as I am preparing it. In manual you will get, that is not an issue, but as of now I will share in everything in Google Drive link because almost all mail IDs I have added. Anyone left who is still not receiving any assignment script via mail? If still anyone is there, tell me.
Sir, excuse me, one thing, just I need to means for clarification, my own clarification. So sometimes in few papers and it has been written as a G block, genomic based block, and in traditional block. So is there any difference for that both?
That prediction we will say, sir. During that time, I will say for genomic prediction, we need to go for that blocks. What you are saying now, for that blocks, we need to go for genomic prediction.
And sometimes it has been also used as I block or I blue. That kind.
Yeah, that each and every block we will see one by one. Tomorrow we will finish that part.
Okay, sir. Another request, sir. Please kindly requesting to upload the videos.
Yeah, today it will be there, sir. Today all our videos will be uploaded. Because of that Durga Puja and everything, these people, I think these people were not there. But today that videos, everything will be there. No problem.
Okay, sir. Thank you.
Yesterday only I talked with them. So because of this Dashera and all things, these people were not there. So they told like today. Yeah, for editing command, wherever is necessary, you need to edit. Like file name, everywhere we need to edit. So editing command is up to you. But whenever you will use different data sets, so take precaution that these whatever commands I am sharing, these are not ready to use. You need to edit. Just keep that thing in mind. Otherwise, you will use the same command, so it will give error. Okay, so now only alpha lattice, then QTL mapping, genomic selection, then D square, heterosis combining ability, then remaining visualization, that is correlation plot, path and a path diagram. So these are our pending part.
Sir, line into tester is there.
Yeah, in that heterosis combining ability, all the models will be covered.
Okay.
Like diallele, full diallele, half diallele, line into tester, everything. When to use diallele, when to use line into tester, which one is beneficial, which one will give what kind of result, everything we will see. And the remaining graphical representations, some representations I have told, but some are pending. So this chord diagram you can use for your RBD data also. No, it will, I don't think, because we have lost some 3-4 days. So again, it will be extended till 3-4 days, Raj.
Okay, sir.
Because we have lost some days, so we need to cover it. Yeah, clustering D square. There are different parts, that is edge clustering, k-mean clustering, hierarchical clustering with different dendrograms. So that also we need to see. And try to submit assignment. The second assignment. Yeah. Path diagram, correlation plot we have already seen. So you can use that chord diagram instead of heat map. I will show you the path diagram, how to draw. For RBD augmented, I will show you this in single script. Okay. So that my laptop issue is almost, I think, solved. So tomorrow that ICI mapping will work on my system. So I can show you. Tomorrow, anyhow, we will complete that ICI mapping, QTL mapping part. How to convert into numerical data already we have seen. Then we need to arrange that data in specific format. So if you have installed ICI mapping, so in C drive, if you will check, anyone installed ICI mapping?
Yes,
Yes, sir.
Yes, sir.
Yes,
So
sir.
in C drive, you will have, if you will check, you will find one folder with name CAAS. So in that you will find some example files are there for 3, 5, 5, 6 crops. So just try to access that ICI mapping software. Tomorrow we will see that part.
Sir, kindly send the link for QTL cartographer.
Cartographer, that link is not working. I don't know what happened. Just
Yeah, for
try.
me also the link of QTL cartographer is not
Yeah,
working.
yeah, cartographer is not working. I don't know why. If till tomorrow, if it is not working, we'll use one binary function via R, that is QTL GCI mapping GUI version is there. So we will use that. I will share that script, how to install it. So if that cartographer will work, so that is simple, that initially you will understand easily. So, okay, tomorrow we will finish that ICI mapping and then we will work according to the status of that websites, whether it is working or not, then we will see. Okay. Just try to submit the assignments as early as possible.
So can you please tell again what did you say about that CAAS?
That folder will be there in your C drive.
Yes, sir, it is there.
Yeah, so in that folder, you will find some examples.
Okay.
Some files like Arabidopsis, rel, maze, .bip file, some map file, BIP file, like that files will be there.
Okay, so we have to go through it. Is that?
Yeah, just go through it.
Okay, sir.
Go through it and check the formats of that file. So likewise, we need to arrange.
Okay, sir. Okay.
Okay. Thank you. So tomorrow we will see First, how to arrange the data, because for QTL mapping, we will use the numerical data, what we have seen yesterday. So again, we will convert that, again, we will use that numerical command tomorrow, and then we will arrange the file, and then we will run the QTL mapping. Okay, we'll meet tomorrow 6:15. Okay, thank you.
Thank you.
So just try for this analysis in your own data, whichever you are working. Yeah, and someone asked me about PAV Jivas. Who asked? Yesterday
It's,
someone asked me.
sorry, it's me, sir.
Ah, yeah. Raju sir, we'll see that also.
Okay, sir. Okay, sir.
Yeah. That PAV Jivas is based on pangenomic data. So that we will that also we will see. Okay, thank you. We will see tomorrow.